I'm reasonably flexible. I can bend my finger back about this far. Some people are hyperflexible though, and this can be as mild as being a party trick where they can bend their finger back to their wrist, but also in more extreme forms, it can be quite disabling and even fatal as their connective tissues and even their aorta become weak and can possibly rupture. This is a whole collection of diseases known currently as Ehlers Danlos Syndrome, and we don't have a very good way to study this in humans.
Ehlers Danlos Syndrome, or EDS, is caused by mutations in genes that produce connective tissue. Among the most common of these genes affected are the ones that produce collagen, which is a long fibrous protein. It's the building block of skin and accounts for about 30% of all of the body's protein. Scientists studying Ehlers Danlos Syndrome use mice or zebrafish to better understand how mutations in genes like collagen lead to the condition.
However, there is an animal model that we look to try and understand how connective tissue holds itself together or doesn't. And that specifically is the connective tissues of echinoderms, so starfish and their relatives.
What's fascinating about starfish is that they have a unique type of connective tissue called mutable collagenous tissue, which can rapidly switch from soft and pliable to stiff, and then back again. And that's not the only thing these odd organisms can do.
Now, you may be familiar with the idea of lizards losing their tail or crabs popping their arms or claws off if they feel threatened, but starfish can do the same. They can break off their arms sort of voluntarily, especially if something like a seagull is pecking and pulling on one arm. And they will grow that arm back later. It doesn't hurt the animal in the long term.
The process is called autotomy, and whilst it seems really dramatic, the evolutionary advantage is that it's better to lose a limb and to live another day than to get eaten. One of the key processes of this ability is to stop the connective tissue from being, well, connective.
Now, several people, including myself, have studied the mechanics of the connective tissue.
But Anna Tinoco and Maurice Elfecht, and the rest of the group at Queen Mary University London,
recently published a finding where they sort of explored a different take on this model. They looked at the neural input to what actually causes the arms to voluntarily break off.
The animal must create a signal that tells the limb to fall off. So how does it do it? Here's what Tinoco did.
She took starfish and pinched their arms in a clamp, and she then injected them with a neuropeptide called CCK1. Now let me unpack that. A neuropeptide is something that would be secreted by a nerve cell onto some tissue.
The full name is ARSK-CCK1. AR is for Asterias rubens, the common starfish, and our model organism. They were already studying this molecule because it plays a role in feeding behaviors. But by accident, they noticed that it also triggered autotomy.
When she pinched the arms and injected the starfish, she would find that the starfish would allow its arm to be broken off or would jettison it. Now again, this doesn't harm the starfish in the long run. It can grow a new arm back. Of those that she injected, over 80% of them lost one or more arms. And when she did the control by injecting some with just water, none of those lost their arms.
So the neuropeptide seems to signal autotomy. Now, the starfish nervous system is quite different to ours. Instead of a single brain, they have a decentralized nervous system. Each arm has a bundle of nerves and can act independently to the others, but all the arms are connected by a central nerve ring. So, this led Tinoco to a question.
Was CCK1 affecting some upstream part of the animal, like one of the 5 brains, or was it directly affecting the breakage site? Well, to answer that question, she made antibodies to CCK1. These are very specific little tags that she could stain to find where CCK1 might be in the animal. She then took non-injected animals and looked at the break sites and put CCK1 antibodies on them. And in this micrograph, we can see that it's stained right along the breakage line, implying that CCK1 is indeed being secreted by cells at that site. Maybe they're still being secreted in other places, but they seem important here. And what else is at that site? The tourniquet muscle. This is a muscle that pinches off right at the break site putting pressure on those connective tissues and helping to separate them from the body.
This work is important because, like mice and zebrafish, if starfish are going to be used as a model organism for studying disease, we need to understand how they work.
So what she did was to open up a new way of looking at this model system in this model organism that we use to study a whole collection of diseases that we call Ehlers Danlos Syndrome. Remember that between the question and the answer are all of the really tasty bits. Hey, SpongeBob, watch me pop my head off. Oh! Starfish actually have eyes. They're just on the tips of their arms, but I think it would freak children out if they built Patrick that way.